talk, I named the Speciation with Migration in the Derrida Higgs model to try to motivate a bit for the talk tomorrow. My research is in speciation models that are inspired basically by the Derrida Higgs model. So I'm going to talk a bit about things remind of spin glasses, but they're not so sophisticated because we have a lot of biology in it. We have to understand something from evolution, from ecology. And so the talk today, I will try to bring these concepts and motivate why it's important to study speciation models and what we can learn from physics models that we adapt to explain this phenomena. I hope that for the end of the talk, I'm going to talk about my results with the speciation with migration. And for that, I will go through the beginnings of this, we call the Derrida Higgs model. But first of all, I don't know if you guys are used to concepts in ecology and evolution about speciation. So I'm going to just give a bit of background. When we are talking about evolution, the first thing that everyone thinks is Darwin, of course. Then his seminal work in origin of species by means of the natural selection. He introduces some concepts by the observation he did on his work in the Galapagos Islands. So one thing that Darwin pointed out is we have this concept of a species that are a group of individuals in biology. They are very like each other, they look alike, they behave as the same, they eat the same thing, they are living in the same place. But it seems like they are not homogeneous. There's always some variability. And not only you have like differences inside the species, but also when they have offspring, some of these features, sometimes they seem that they are passed more frequently than the others. So another thing is that you have inheritability. But this was not occurring in the homogeneous way. Some features seem that they were more prevailing than the others. So one thing is, the species, they are not permanent. They are evolving, and some features, it seems that they were being selected. That's the word that he used. So in fact, that's why he introduces the concept of the natural selection. And the idea that species are evolving. These groups of individuals are changing over time. So when you have that sketch that nowadays we use, that was the tree. So we have, over time, this group of species that are changing. At some point you have, they divide into subgroups, and they continue to change. In a point that these two groups, they not identify each other as a species anymore. So in some point, you have what you call the speciation event. So this is something that we have the idea today of how we describe the history of a species. They all come from a common ancestor, and you have these events that produce different species. They split into species that we call the speciation. But when I start thinking, like, I have the groups of individuals, and at some point they are not identifying themselves as the same group. We are using a concept here. So the thing is that we have to define what is a species. And for this, we call another zoologist, a bit after Darwin, Meyer, he wanted to study this kind of thing, that the study of species we call systematics. I want to catalog the species. So we have to define when is this point, where is this point, and then we use the concept of reproductive isolation. So in fact, these groups, the idea these groups will become different species when they are not interbreeding anymore, or they don't even have the potential of breeding. This is what you call the biological species concept, is the one that's more prevailing, let's say. But species per self in biology is not so straightforward, because if you think you can have, for example, living beings that don't have sexual reproduction. But for so, we will continue with that. We'll see how we can later make a more broad concept of a species. This is for now. We can concentrate the ideas that we wanted to explain speciation. So one thing is that we had the natural selection that was acting on the variability, but then after, in the 20th century, we saw that everything that is behind all of this is genetics. So we have to try to explain things in the microscopic level. So in fact, when you want to describe that variability, in the genetic levels, they come from what we call the evolutionary forces. So Darwin was observing the phenotype, what the features that you can observe in the group of individuals. But the thing is that microscopically, in more underlying level, what we have is the genetic population, like what the genes that the population have. And that's where variability starts. So when you describe the evolutionary forces, is what makes the produce variability in the genetics of population, that in the last level we produce different phenotypes. So with the advancing of the population genetics, we can then englobe these things that produce variability. The first, of course, is mutation. Then when we have the reproduction, we produce gametes, you may have errors of copy. So this is the major source of variability. Then we have something that removes variability. Also, during this process that are inherent to the production of gametes, when they have the copy of genes, you can have by chance the elimination of some genes. That's called genetic drift. So these are two things that happen by chance and either produce or remove variability. So, and of course, then after that we have the natural selection. That change the frequency of these genes. So once you have this pool of genes, the ones that are going to be more fit for the environment, and you can also have the move of genes. We call gene flow. That's what we're going to talk later. That's migration. So this is all the things that change the variability of your population. And over this, the species are going to go through. Another thing that we saw is that I explained to you, like, we have one group, and then some point it becomes two groups. So one thing that englobes everything here and make this becomes two species is geography. Remember that at some point the populations are in the same environment and they are under the same selective pressures, and then at some point you can have a split of the population. So one simple way of thinking that speciation will occur is once you have one cohesive species, somehow this is split into two subpopulations. And then here, these things occur and change the genetic frequency of here, such that you have here these genes and here G's genes. And then at some point when they put them together, they don't identify themselves as in the same group. This is the most simple way of understanding speciation. And that's what we call, when you have this physical separation of subpopulations, that's what we call allopatry. And then, when you think about a physical barrier, could be like geological time, but it has been proved like that you can have a fast speciation, like really fast, some generations.
Just a few from few generations?
Yeah.
Okay.
For example, the birds of Galapagos, some lizards, few generations you can see speciation occurring. So yeah, like I'm going to talk like it's a lot of ingredients. In fact, biology is leading with a range of the phenomena can occur. Like you can have things that are species that are very resilient. They are like almost the same for millions of years, and species that are evolving so fast, and they are splitting so fast. So that's why we study models to try to understand what are the main factors that are dealing to that. So yeah, that's a good question. Like this is like millions of years. So for example, when you think about islands, you can have like you have the structures already there. Like the birds, for example, they're gonna migrate between islands. The physical barrier is permanent. But for in some parts of the Earth, you can have the sea level changes a lot. So you can have generations that are separated and some that are not. In fact, this is my call for what I want to say. This is not like always like this. Sometimes you can have some exchange between individuals here. That's the gene flow, migration. Sometimes you don't even have the barrier. Or you can even think that if you have the physical barrier, you don't need even include the natural selection. Just thinking that you have these things that always are happening in the microscopic level, this speciation will occur anyway. So you have several elements that can be present or not. So the idea of building speciation models is understand what is the effect of each one of these ingredients, and how they can hinder or promote speciation, and what are the effects that we can see, like if species are long-lived, if they are super abundant, and this is mainly what I do in my research.
And the fact that just one of the two branches you did this change is because in the new geographical environment, this change was advantageous, and in the other geographical environment was not, I guess. Like,
yeah,
you know.
That's the under the natural selection, yeah, you can do that, but you could have environments that in fact are like the same conditions. But once they are separated, they are going through changes that are not being exchanged between them.
No, I understand, but why in just one of the two branches you do the change from the cracks, from the X to the just...
Yeah, no, I just wanted to show like that, okay, this the same in fact, like this remain the same species, and this is a subpopulation that went to another environment.
Yeah, and in that new environment, it's convenient to make the change.
Okay, you can think about it. If you have selective pressure there, then okay, this gene was more
convenient, let's say.
Was fittest. But sometimes it can be the same environment. But here occurred a mutation that was by chance went through.
Okay.
That's the point. Like there's some. This is our argument in evolutionary biology. If natural selection is that important or not. Of course, there are some cases that is really clear that the populations are under selective pressures. But, for example, this very long process of speciation, not necessarily. They are just accumulating mutations that occurred here and not here.
I see.
Okay, so the thing is, the things that we can really not get rid of is this one. These always occur. Natural selection, most of time, but sometimes you can think of phenomena occurring and they are under the same selective pressure.
No, because I would tend to think that natural selection is something that you can model with the minimization principle. Instead, the other things are...
Yeah. In fact, one thing is that we're going to talk about models, and the idea is started to talk about microscopical models. One of the things that they use the idea of natural selection is the landscape, the fitness landscape. So you have some, like this is genetic space, you know that you have genotypes that have higher fitness.
Yeah.
So, but the thing is, sometimes you can have not, this is an idea that you are always going through an optimal, but you have fluctuation. This is not always true. You can have a population that sometimes, for example, accumulating deleterious mutations, they're going to skip this optima. So this is also something that is important. Not natural selection is important, but you always have to account for those things that are creating randomness in your system. So in fact, this was a thing, like when I was studying against these things, I remember that studying genotypes in the fitness landscape, and that was a problem. Like if you don't consider the randomness, they are important. Otherwise, it's just a minimization.
Yeah,
There is no intention in going to an optimum. Things, even going through this peak has to happen like by random chance, like to have the mutations that are good to reach that optimal fitness. So, with this, I hope I got to motivate why it's important to considering stochastic into genetic modeling. And this was what Higgs in Derrida, also with Peliti, and what's the name? Sorry, I forgot. Serva. Around the 90s, they were developing this spin glasses model. And by the time, as we know, like now we have the Nobel for the spin glasses with applications in complex systems. We know that these things, they have applications in several areas, especially when we are talking about phase transitions. And the idea they had is, once we were talking about something that's microscopical, there's a lot of stochasticity involved. What if we treat the genetic space of a species as a spin glass, and then speciation is a phase transition in this genetic space. And this is almost straightforward. So the idea is we have like spin chains, Eisen spins. We're gonna use them to emulate a population. So we're gonna call that you have an individual. It's gonna be represented by a chain of spins, and this will be its genome. It's a very simple way. Of course, we now know that genomes are very complicated. We have the bases, we have proteins. But what we are saying here, we're incorporating the idea that we have the genes. Each spin is a gene. It has like two values. So it's like a gene that has two possible alleles. They are independent. And they're somehow going to describe your population. So we're going to start this description. So one thing from these elements, remember the two things that we are always going to consider, mutation and genetic drift. So we're say, choose the ingredients. So we're going to consider the flat infiniteness landscape. So, okay, we're going to model a population. Individuals are going to be spin chains, and there is no optimal genome. Like, they are all equally likely to survive. There is no, like, -1 and +1 in each position is equally good. That's also what we call a neutral model. But here, when you mutate the gene, we're going to switch the value of the spin. It's not going to be good or bad. It's just a change. And first, we're going to see what happens when we don't have geography. Meaning, okay, gonna start like from the simple idea. You have individuals. Each one is described by a sequence of spins. They are equally like to reproduce, and we're gonna put some rules here for the reproduction and see what happens with evolution. Okay, so, and with this, we're gonna see if you have a phase transition here. It's gonna be purely by effect of mutations and genetic drift, okay? So the first one is the okay, even easier. If you don't need to mate, we just produce clones of yourself, like your bacteria. We just, like saying bacteria that are in a petri dish, we just produce clones. How are we gonna evolve? So we consider a population of fixed size M, and that's how you're gonna evolve in the generation. T-1. We have a guy, he's going to produce an offspring to generation T. So our model is going to be with no overlap generations. So each, at one generation, we're going to kill all of the population and replace it by the offspring. And here, so we have the genome of this guy, and here is the genome of the offspring. And since we are doing cloning, we just copying, but from the chance of mutation. So each of the genes of the offspring can be either equal to the parent or switch. This is the mutation. Once you say that mutation occurs with probability mu dt, so each in interval dt, it occurs with a probability mu. In fact, from one generation to another, this will occur with the probability 1/2 plus, and this so for each parent, we have a description of the possible offspring with a probabilistic description. And then G is just parent. It's just the parent. I just use the name G. Could be P of parent. This is the parent producing an offspring from one generation to another. And the genome of the offspring is equal to the parent, but from mutations that occur with this probability. So we have a probabilistic description of the genome of the offspring. One thing is that is going to be important then from us is to describe what we call the similarity between individuals. We'll call the genetic similarity between them just by the product of their spins. So if they have, so in the correspondent sites, when you compare the two chains, if they have the same value, it sums up to a positive similarity. If they have opposite signals, it shows that they are less similar. And with this, because we have this description, we can calculate what is going to be the expected value. From one generation to another. So basically, just calculating, we have what the spin of the individual alpha is this probability of having this one, or this probability of having minus this one. So we can build the similarity of the offspring with respect to the parent's generation. If we do this calculation, we'll get something like this.
Sorry, just to understand the notation. What you call alpha and beta, if I'm still understanding, I would understand more like S, I would call it QP alpha alpha. Am I right? I mean, you are comparing the parent of alpha with alpha, am I right?
No, here I'm comparing two individuals in the same generation, alpha and beta.
So are you assuming that the two parents, the
no,
parents
yeah,
of alpha, and
I mean, so you have beta and you have alpha and the parents of alpha, and beta and the parents of beta.
Okay, but so the initial distribution for all the parents was equal to everybody? Because, I mean, I find hard to compare two guys that are coming. I mean, I would understand the comparison between the parents and the son, or the parents and the offspring, because you see how frequent you do changes.
Okay, so
but
okay,
I'm
maybe
a bit in trouble in understanding.
I can explain better. The thing is that we are doing non-overlap generations, and we want to analyze the structure of each generation. So we're going to say, like, how, if you start from clonal individuals, like you start from individuals that have the same...
that was my original question.
Yeah.
So you started from an initial configuration in which everybody has exactly the same genome. Okay.
Yes.
Okay, great.
Yeah, it's because I'm going... Maybe I was confused here. Maybe I should start with explaining the dynamics. No, yeah, that's okay. Because this is just like a measure that we're going to introduce. So to each generation, we have to use some metrics to identify, like, if we're going to see something going on. So we start from, good. Yeah, we are just starting from population that are clones, all the same. What happens? So once you have the parental generation, how we can compare to the next, the offspring. So we compare two individuals in the same generation using this, and then because we have this rule of how we're going to have mutations, how things are going to change, we have this thing that in the end is all this just to say, if you have purely mutations, the generations are changing because of mutation. And the similarity is decaying because mutations are accumulating. And why I did this? Because we didn't say anything yet about how long is your genome. And one thing that when they proposed the model is, let's start with the simplest way, that is, if you have infinite genomes, in fact, this matrix, they are, the fluctuations are negligible because the fluctuations go with 1 over square root of n. So these things we can deal like not only expected value, but is the rule of evolution. So we have how generations are changing. If you have infinite genomes, we know that once you have the similarities of one generation, the following generation is going to be like this. And not only that, once you said, like, if you start from clonal population of individuals, you see that in the beginning, this is going to be 1. So any term, when you have a really split of things, it will appear just a term here that gives you the time of divergence. Then one thing is that once you do this calculation, they did, what's going to matter then is this distribution of Qs. Is the so we in the diagonal think that this is a matrix that's this is M by M matrix that describe the similarities in your population. In the diagonal, you always have 1 because it's the individual similarity with itself. But then what you want to observe is what happens to the non-diagonal elements that's going to follow this rule. And then to do the simulations, we have to add the other elements, that's the genetic drift. What it means. Remember when I said that the parents are going to generate offspring? If you want to introduce randomness, in fact, not all of them are going to produce. You have to do by chance. So to do the dynamics from one generation to another, we say that each time that we want to replace to generate the offspring, we have to draw the parent from the other generation. So in fact, if you think, if you do M draws, each time each individual have 1 over M chance of being drawn. This is the chance of not being drawn. In M draws, to substitute all the population, this is the chance of individual not to be drawn. So when you produce this new generation from the population, in fact, some individuals won't produce offspring. And this is our element of the genetic drift. Some parents, in fact, are not going to produce, and some are going to produce more than 1 offspring. Okay?
But so the number of individual is fixed?
Yes,
or,
this is fixed.
Okay, so if you don't generate, you don't die. If you generate, you die. This is the rule. Okay.
At the end of the generation, everyone dies. But then you do this M draws to produce the new population.
Right.
And at each time, you always have the chance to being drawn.
Okay.
And when you do the simulation, what you get is that you have some distribution of peaks that, in fact, move with time. They don't reach a steady state. This continues to change whenever you following these rules. So this is what they. Sorry, my board is horrible.
On the horizontal axis, what What is there?
Here you have like the values of Q and the distribution. So for example, when you look there, how frequent is in this matrix this value of Q? How frequent is this value of Q? So we have groups that are some groups that are more similar to each other than others. For example, you have groups that are super distant from each other and groups that are very similar. So kind of, even if we are dealing with asexual reproduction, we don't have that crossing. We kind of have that idea. We have groups of individuals that are more similar to each other. So we kind of have a definition of species going on here. Sorry.
So I didn't understand. What is the rule for selecting the number of offspring a given parent creates?
You always replaces M. You generate M offspring, always. But then everyone in T minus 1 is dying. And then to each offspring that you have here, you're going to do a new draw.
And you draw how? Just at random?
Random.
I see. Okay. So you have the set of new offspring, and their parents are assigned
Yes.
at random.
Yes.
Okay. Thank you.
Yes.
I'm sorry, I'm a little confused. So SI alpha, the index I indexes the length of my genome, and the index alpha indexes individuals.
Yeah, this is the number of genes, this is
Good.
the position of the genes, and this is the individual.
Good. But then the update rule says that from one time slice to the next, each one flips independently of all the others, right?
Yes, they're independent, yeah.
So that means I have a whole bunch of independent Ising. If I think about the time axis, then I have a whole bunch of independent Ising spin chains, right?
Yes.
So tell me where that problem, I don't get non-monotonic distributions, do I? I certainly don't get a phase transition. So I'm somehow these must all be tied together in some way that I'm missing, or maybe you haven't told us there's a phase transition yet, I guess.
No, yeah, one thing is that when I include, when I said to you that you have this, in fact, if you think like the expected value, it seems like it's only decaying, right? But when you include the rule of, when you do the simulation, and you include the rule of that you have, they replaces by random, and the simulation, the result is that you don't have a steady state of the distribution of the matrix.
Sorry, I'm trying to reconcile the rule of replacing by something random with the thing that you wrote at the top left there, where either you stay the same with one probability or you flip.
Yeah, because the thing is that in the end, we're not going to simulate the whole genome. Because what they wanted to calculate, when they suggested using the similarity, the thing is that we only using the similarity between the genomes. We are not simulating the genomes per se, but the similarities. What matters to us is how the individuals are similar to each other. So in fact, what we are building every time is this Q alpha beta. And here is where I include this. So this I know that each spin of alpha has this probability of hitting the parent or this being with the switch. And we have this. So now, okay, now I know how is each element of the matrix. And then to each element of the matrix, I have to compare how the parents are related. So sometimes individuals will have the same parent. So for example, if alpha and beta have the same parent, this is going to be 1. Yeah, you can have that. Yeah, yeah, no, yeah. Then it's gonna be trivial, yeah. Yeah, that is the thing that we were inputting the thing of that some can have 2 or more offspring and some don't. That's gonna generate some mass there. And sorry.
Yet we're not putting any pressure, any
No.
kind of external mechanism that suggests that somebody will have 2 sons and somebody will have 0. It's
No,
just
yeah,
random.
no,
Okay.
yeah. That's what I say. Okay, I'm starting really naively, and we're gonna progressively include these things. Yeah, no, this is really, really naive. Like, we're just considering asexual reproduction, just mutation, genetic drift, nothing else. And even then, we can see that there is something going on here that doesn't reach a steady state. So we have this. No, yeah, I'm just random here, but yeah, you can have like 3 peaks because you have like some individuals that are very similar, some pairs of individuals that are very distant.
But ultimately it comes from who your parents are, right?
Sorry?
Doesn't it come from who your parents are?
Yes, yes. Well, this is like after several generations.
Okay.
A lot. Like thousands of generations. You can know how long this takes to appear based on the size of the population and the mutation rate. Okay? But yeah, like if you think like count the number of peaks, you have a long time with just 1 peak, and then you start to have some.
And the number of peaks is somehow related. It's how related we can be, right? So
Yeah, remember
if
that
if I look at a pair, alpha and beta, that are the offspring of of completely independent parents that never had an ancestor in common, then I get one thing. If they had one ancestor, a recent ancestor
yes,
in
how
common.
long
Okay.
their ancestor are, like how long their ancestor was, yeah.
Sorry, and you claim it's reproducible, so it only depends on mu and m. There's no randomness.
To know the time that will take to these peaks to split.
You're saying that the number of peaks and the time it takes to split is just a function of mu and m?
Yeah, I can. Yeah.
So it's reproducible. You do it 100 times with the same mu and m, you'll get the same answer?
Yeah, of course, rather from variations, but the average, yeah.
Okay.
Yeah, in fact, we can because here we don't have the concept of species yet, right? I can, like you just already put here, that, for example, we can use a criterion that everyone that has similarity higher than this is the same species, and one that's higher is low.
So you have a function of mu and m, which tells you how many peaks you'll have and how long it takes for them to separate?
Then you have to look more detailed into their work, because I'm more interested in number of species. And for that, I can have, later I can have I estimate.
Okay, so there's a function. You can just write down a function.
Yes, you
Okay,
can have.
thanks.
Because, yeah, that's the thing. Where is it? Here. No, this is already done. Here. That they showed in their work is that so we call this distribution non-self-averaging. And in fact, when they calculate the time average, they got something that's like, and this lambda is 1 over. Sorry, it's difficult to.
So the fact that it's non-self-averaging means that it varies a lot between different runs, right? But something about the number of peaks it has would be more, each run would have a typical value for the number of peaks at a certain time.
Yeah.
Because non-self-averaging means that P of Q is completely different each time you do a run.
Okay, no.
That's what non-self-averaging means.
Okay. Yeah, that's right, yeah. Yeah, but again, it's because I'm not interested in the number of peaks. I'm interested later in the number of species. So then I will have an estimate for that. So, okay, I get your point. Yeah, of course, every run is going to have like different distributions because it's random. Okay.
Because it's not self-averaging, right?
Yeah, yeah. Okay, but, well, so as I said, like, there is some properties regarding this, but yeah, it's not no self-averaging. So the thing is, even with a simple thing, we see that there is some kind of phase transition going on. So next thing they did is, okay, let's include then sexual reproduction.
I start with, I think it's related to this notation that I'm confused of.
Sorry.
P of alpha going to alpha instead of alpha going to sun
wow.
of alpha. So I'm a bit confused by this one to many and how time evolves in one direction. So if I have one individual, then at the next step, it splits into a bunch. That's true? That's how I should think. And then each of them has those spins that can be similar to the ones above, up to variations. And then it repeats, so it keeps opening like a tree. But I'm confused. That cannot be the correct picture because they are talking to each other, I assume, right? So I have a bunch of individuals. It's not like one of them starts multiplying and dividing, and I ignore the other ones. So how are the different?
They are ignoring the other ones because it's asexual reproduction. You are just producing clones.
So when we say I have many, okay, I can.
Yeah,
I ignore the other ones. I focus on one and I repeat many times. It's about one spin splitting, and studying what happens.
Not just splitting. When you have one generation, you have here alpha, beta, beta, gamma, and then in the next generation, this is the parental generation, for example.
But alpha, beta, and gamma don't talk to each other, right?
No.
So why do I draw them there? Why don't I just put one dot? Why am I putting many dots if they are not talking to each other? That's what I'm confused. Why am I putting alpha, beta, and gamma if they are decoupled?
This is one generation. Like, I have to describe this generation. It's okay. The next generation, how it's going to build? I'm going to choose randomly here, and this is going to be the offspring of them. But they don't need to talk with each other. Like this, for example, can produce two offspring here, beta 1 and beta 2. And this one can produce gamma 1, and this won't produce offspring.
There is an effective interaction because there's a constant population constraint.
It's a constant population, always M. I have M individuals here, I will have M individuals here.
Okay. So that's because there is some limited space and they occupy everything.
Yeah.
Okay, thank you.
We are in the carrying capacity of they are occupying, they have competition for food. So yeah, you always have the same population size. So you're always producing each generation M offspring.
If you choose an order, like you start from alpha, then alpha has somehow more freedom than beta, and gamma has zero freedom. Because, for instance, for example, suppose that you start doing your sample from alpha.
No,
Alpha can choose reproduce twice.
Schematic. I don't have any space. I don't have anything.
No, but in any case, you need to scan all the individuals and decide how many offspring they do. But once you choose an order, that order determines that.
Oh no, I don't choose in order. Like when I produce.
How you do
that? For example, I'm gonna say how I do the simulation. Okay, I have the first. This is the first generation. I have an index for each individual, right? The second, I say, okay, this first individual is gonna be. I do a random draw. It's gonna be the offspring of individual 1.
Okay.
This one is going to be the offspring of individual 1. This is going to be the offspring of individual 3.
Got it, okay.
Okay, and then, okay, this individual 1 these two, they are the offspring of the same guy. So I just put the A to the here. Oh, yeah, yeah. Sorry for that. And then, well, I think I got it now, okay? That's why biology asked, like, how it's the structure of this thing. It's because of that. Like, and this thing is not in that update rule. Like, okay, so now we want to. Good that you resolved that, because it can get even more complicated when you talk about sexual reproduction. And now then each individual will have two parents. So when you look at alpha, it has parent 1 and parent 2. And beta has parent 1 and parent 2. Oops. Again, they're chosen randomly, so whenever you build the individual 1 in the first matrix, you choose like 2 individuals. First you choose 1, and then you choose another one from the ones that are left. And then you can do that calculation again, following the same idea of mutation. And then what you got this time, because you have these possible things. Oh yeah, one thing is important. When you talk about sexual reproduction, we think about recombination. Right? But to make the model simple, what do we have? Like, instead of just copying the gene of the parent, you follow this rule, but with half of chance of copying of the parent 1, or half of chance copying of the parent 2. So in fact, you have like for each gene of alpha, you have 4 possibilities, right? Is this would have chance for parent 1, then this for parent 1, and another copy of this for parent 2, each one with half of chance. So giving all the possibilities, we can open that that matrix, in effect, with a similarity matrix, and now it's going to be like this. And all the combinations of parents. I mean, now I also have the thing that each gene can be being received from each parent. And the thing is that when you do that, and you look at that distribution P of key, after a lot of time, now produces only 1 peak. It reached the steady state. So somehow this sort of recombination is producing some mix that makes that thing goes away. And in fact,
This, or is it just an observation, or what?
Again, in the simulations, you observe that.
Simulation is an observation. I'm asking, can you prove it? Is there just some way to prove
We can
that?
calculate that. I'm going to calculate that. So we can calculate this peak. And that's it, how we do. So how we calculate that? We want to see how is the average, population average, of a value of Q given two pairs in the next generation. If you get two individuals, they have two possibilities. They have they can have one parent in common. And this occurs with probability. And of course, you can calculate this but this for large M, okay? Just do the probabilities for large M. This is the probability. Or they can have no parent in common. And then the probability is going to be you see that because when I build this this average, these are the ones that matter, because if they have the same one, the two in common. Oh yeah, this is very small. When M is large, it's gonna be very small. Yeah, I'm making an average. So the probability of having the two in common, you have to do the draw. I can make it here. For now, believe me, when you have like a very large population, two individuals have the two same parents is a very small probability. Yeah, yeah. Okay, thank you. So when they have one parent in common, their similarity is gonna be like this. We're gonna have that term that E minus to the minus 4 mu over 4. One of those terms is 1 because they have one parent in common, and the other 3 is just the average of the previous. Okay? When they have one parent in common, we are producing an average. Okay, we're just average things. Like if you want to calculate using this thing, a single one of these terms is 1. All the other ones is in the average of we are putting the average of the previous generation. And if all of them are different, in fact, then they're just gonna change by, because then it's all the same. It's just gonna be like what you had before, just e to the minus 4 mu. From this, we can build these averages with chance for M, this thing. And with chance this thing. I see, sorry. Huh? it's just that, yeah, because this is the probability of having one parent in common, and this is no parent in common. Huh? Ah, sorry. Yeah. Well, this is gonna give this thing like this. And then we can try to think in a possible steady state, and then we get something. That's where the peak is. So when this average is gonna reach a steady state, we're gonna reach this peak. So well, I'm short in time. I'm sorry. I took so long. Yeah. Well, so then to recover speciation, then we have a very simple idea. In fact, we have the thing that the population is reaching a similarity, and then all of them are weakly distinct by this term. So you don't have a species. One element that we introduce then is making this reproduction selective, in a way that only individuals that are a bit more similar to each other. So then when we choose these parents, they're not random. They can only reproduce if their similarities are higher than a limit. So we're introducing, okay, not all of them can reproduce with each other. We produce a limit, and then we put this, we recover that. We recover the species breaking. And we can think that what happened then is once you have this, so we have the limit here of Q. And now we are having that thing that I told in the beginning. We have a criteria for reproductive isolation. We can calculate the number of species. For that, we do this approximation. Given that the peak of when the population is size m, the peak it reaches steady states in Q0. We think that given a species of abundance m, it will be stationary at that abundance. So using this, you can calculate the average abundance of each species. Given the total population, the number of species is just divide the population by m. Okay, I'm sorry that I wanted to talk about migration too. I don't think we can make it. But basic is like another, the next element is, now we talk about populations, and you're gonna allow individuals to be exchanged. We have population M here, population M here, and before the reproduction, we have each individual will have a probability epsilon of changing the islands. We have 2 islands model here. And we want to see what happens here. Again, we're going to use the similarities to describe the population. We have now similarities inside each island and between different islands. So I'm going to just put directly here. So this, the probability of the two individuals changed island, or the two did not change the island. So they were in the same island before, and they remained the same island, or they both switched islands. So it's going to contribute with the factor, and this Q here is basically this term here that I just opened. And for individuals that now are in the same island, they could also be in different islands before. If they were in different islands before, for sure they had different parents, so the contributions is e to the minus 4 mu, and this probability is this is the probability that one of them switched island and the other one didn't, so times 2. We can build the same for the similarities between different islands. So if they were in different islands, they remain in different islands if they both migrate, if they both remained. And if they were in the same island before, then there are all the possibilities of they having the same parents, blah, blah, blah. But one of them switched, and one of them didn't. So they contribute to this similarity between different islands. So again, I can also get expressions for P naught and Q naught. Well, then we can get a look in the paper. The important thing is that we have an expression for the number of species. So we're gonna define now these two things. So we have always the similarity inside an island is always going to be larger than similarity between different islands. And we can notice that when the migration is very large, in fact, when migration is too large, like this flux is very large. In fact, this system is like just one system with two population. So with this, again, I can use that trick to calculate number of species, and we get something. Like number of species per probability, average number of species per probability of migrating. So the number of species in each island increase with migration, but the total number of the species of the system, this case. And why is that? When you have migration, what's going on is that there is some exchange here that's going to promote speciation, it's going to promote diversity. So we have what we call the gene flux conundrum. So we think about number of species per dispersal. We have something that's like this. Like, a bit of migration is good, because remember that you're bringing some gene diversity to the island. So a bit of the migrants arriving here, they are bringing some diversity. So it's increasing the number of species. But at some point, the species start to be shared between two islands. They start to have the same species. When the islands are isolated from each other, the species that are here are not the same as here. This is what we call the endemism, the situation of max endemism. Like, that's when the number of species, total species, is maximum. But once you start to have migration, they start to share some species. But in each island, the number of species increase. So we have this thing that we study in ecology. Some migration is good because promotes diversity, but when you have too much migration, it diminishes diversity because they start to mix too much, so that you end the endemism. So basically, we use this model to understand the mechanisms that these occur, because using the simulations, we can understand how these genes are promoting speciation and what are the modes of speciation that are occurring. And in fact, we see that there are two mechanisms. For example, we can have always a group of migrants that arrive here, and they don't mix with the residents, and then they found a new species. We call this founding speciation by founding populations. Or you can have the migrants that arrive here, and they can mix with the population that was already here, and this also promotes speciation because the genes here, in fact, even if they were from the same species, they were slightly different. So this is also promoting some diversity. And yeah, with that I think I'm okay. Sorry for running the end, but yeah. But the idea was that this is the base of the model. Today I just make calculations. I'm sorry, tomorrow I'm going to be only simulations. That I'm going to show how we can put other things in this model because in the end, I'm more interested in watching these things and comparing with things I can see in nature. So I have a model that works, that I can see that we have species breaking, and I can regulate each element is important. And in the end, I'm interested in knowing how these species are, how they're connected, like the relatedness between them, how the trees, like, okay, now I'm seeing species being generated here. I want to build a phylogeny. Maybe if I can compare for a real phylogeny, I'm going to see as a signature of this happening in nature. So, yeah, I think that's it.
I didn't understand your last point here, that the eventually higher migration causes less diversity. I mean, it seems like they're both monotonic, right? The last cartoon curve you drew has a peak, but the two curves you drew that show the number of species per island and the total number of species, like migration always helps the species per island and it always hurts the total number.
Yeah, maybe it's not good here because this is like in fact, something from that we get from theory from ecology, because it's not clear when migration is good or not. Like, we want to study migration because of this. We have this idea: a bit of migration is good, that produces diversity, but too much migration, you're gonna mix the populations. It can hinder the migration, can hinder speciation. One thing that I could not show here is that we have another version, because this model, remember, I'm considering infinite genomes. At some point, we also start to work with finite genomes. With finite genomes, we cannot use these calculations. We have to really simulate all of them. And what we see is that with finite genomes, speciation does not occur. So in fact, with finite genomes, the number of species per island is just 1. But we see that migration makes the number of species increase. But when migration is too large, the population becomes too large, it drops to 0.
Sorry, out of curiosity, how much of this has ever spilled into social sciences? If you replace species with the social classes, and so on. Has anybody ever thought of applying this to social mobility and so on? No, too crazy.
No, yeah, like maybe spread of opinions, maybe like you think like we related to people who have a more similar opinion to you somehow. But I'm gonna talk like people like more sophisticated models for that, maybe like. Yeah, whenever, like, this is from my experience working in evolutionary biology. I like the analytic model because I can have predictions and I can have, like, the curves to compare. But in biology, like, we talk about infinite genomes. It's weird. So you have always to be restricted. And then talking about finite genomes, like thinking about people that are described by finite genomes, then this is, we can have models on that, yeah. There's a model with that, with polarization. People described by chains of spins. But again, like you use the infinitely many genome sites because we can have all these predictions. And then, but when you have to adjust to real world, you have like, in fact, most of the time, you have very few genomes being selected, very few genes being selected. Yeah. But yeah, there is someone who applied this model for
I guess I can't quite do it in real time by looking. Can you tell us, is there a regime in which, in the simplest case, even without migration, you can get lots of species coexisting? I mean, I think I'm following how you get a few.
Uh-huh.
Can you get lots?
Yeah, well, we have like the number of species is always controlled by how restricted is your reproduction. Like the more restricted, more species you have, for sure, and by the size of population, and by mutation rate. Increasing mutation rate, more species.
Okay, so there's a whole community of people who are worried about the persistence of diversity in complicated Locke-Volterra models. But it seems like you have a completely neutral model except for a restriction on sort of similarity even for reproduction. So should I not be worried about the persistence of ecological diversity?
Oh yeah, no, sure, yeah, of course. This is like, and then when we want to, as I said, like I want to get these things and compare to real world, right? At some point we're gonna say like maybe to some populations, it's the only limitation is by the carrying capacity. Like you can imagine that these species are occupying different niche. They are not competing. So, and there is a colleague working in this kind of thing. Like, then what if these species have ecological relations between each other? So they are competing for things. Like, of course, then maybe we have the reduction of number of species. Yeah, this is like our no model per se. Maybe tomorrow I'm going to present like my other version, and you see like when you put other things. For example, remember that I get how we build phylogenies. And one thing is that interesting that when you change the elements, these things change. Like how, like when you have a neutral model, then you have a lot of species there. You have here like a lot of species. Like, and then when we put like some selection, you reduce this a lot. So I don't know if no, I get your point, of course. This is a very naive model, in fact, yeah.
I mean, I guess from the point of view of the Locke-Volterra models, in a sense, there's a fine-tuning here that everybody's equally fit.
Yeah.
And so then maybe persistence isn't surprising because there's nothing selecting against.
Yeah, if you had a Locke-Volterra with the J's all 0, so there's no interaction between species, they would just grow to carrying capacity and sit there.
Good. Okay, thanks.
Yeah, yeah. The thing is, like, we can in this system, we are what is defined the number of species is these things like this restriction, mutation rate, and the size of population. Like, and then you can run a lot of simulations. You're gonna get always the same size of number of species. So, yeah. But yeah, tomorrow I'm gonna present the work that I put some kind of selection. Then you see these things drastically change.
I have a question that's more technical. So this method you have of estimating the number of species from the position of the peaks, does it seems like it depends on an assumption that the variation within each species of the genome is the same for all the species.
Oh, yeah, no,
That there's a distribution for each species which is identical.
There's a distribution of abundances, for sure.
No, but I mean that...
Not all species have the same number of...
No, I don't mean that they have a distribution of abundances. I mean that, you know, one species might have a very tight distribution of mutations within it, whereas another species might be much broader, and this would, I think, warp this estimate, no? Because it would make it would change the height of the queue, for instance.
Okay. In fact, when we make some more sophisticated estimates, we consider we have the abundance, but in fact we have like a distribution. In fact, not all species have the same abundance, and you have the variation. So, yeah, about the width of the peak is something that my previous supervisor was working on it, but okay, well, if you think that, well, more populated, but again, like this distribution of peaks is not, they not correspond to one species, right? They are similarity between different species. That's why sometimes it's complicated to mix stuff, and that's why I make these estimates. But what I can say is, like, we have to consider that we have, in fact, a distribution of abundances. Like, we have, like, any species, you have, usually you have this kind of thing. You have a lot of species that are very low populated and a lot of species that are very abundant. Okay? So we have, this is like a mean value of abundance to get the number of species. But it works for approximation. You mean like this, in fact, you don't reach this value of, is the mean of the peak, right? You have a width. I feel like it's somehow related to the fact that you also have variation of abundances. I know that when we do this calculation, we always get a number that's a bit lower than should be. But yeah, we should make a more sophisticated calculation, considering that the peak has a width, so, and also this abundance, in fact, has a width, and then you have a more precise number of species. But this is like estimating, like, and kind of work. I think I don't have the slides here, but you see, like, when you do this estimate, the number of in the simulation is like I don't remember if it's a bit lower or a bit higher, but kind of works. But yeah, the physical statisticals of that, in fact, is much more complicated, and I assume I don't have the tools to do that.
Sorry, I think since I was not here in the beginning, if you said it in the beginning, just tell me and I'll watch after. But was this an attempt to solve some open problem? And what was the open problem that was solved, or what problem are we trying to solve?
The motivation at the beginning was kind of explain what is an speciation, what is a species, and there's this open problem in evolutionary biology to understand how the species they come up, because we want to understand how they appear, how they disappear, how long they stay. So we have to have speciation models. And in the beginning, I showed that to understand how species appear, in fact, there's a lot of elements. We are used to like the idea of natural selection, but there's the whole genetic things going on, like, and then we have to understand like how mutations, genetic drift, how the frequency of the genetic alleles in the population change, and we also have the geographical factor. So we have in nature, when you observe, there's a lot of variation of speciation process. So you want to understand, for example, when I have, this is what I have when I look there. And then I want to see, it's like, okay, this is very likely that happened to a population that in fact was living in contiguous spaces. Okay, can I differentiate in which process natural selection was really important? Can I differentiate when migration is hindering speciation? So the idea is having models that give us the intuition when each element is going to be important or not.
Yeah, I guess I have a similar question. I don't know if this is the right time to do it, but sometimes the theory is, I mean, the things are not well defined enough that you can actually write down a model. I mean, it seems here that the results are so almost ambiguous, or depending on which type of speciation we're talking about, that maybe there is no model that gives the answer. Maybe it's just some, intuition you get from thinking about it, but not writing down some mathematical.
Yeah, sometimes you get intuition from your models.
No, the other way around. Maybe you don't need the model, you just need the intuition.
Let me put it the other way around. There is one paper from Science when they did a large simulation, when they simulated the world. They put everything, geography, climate, competition, niche competition, and then they get how the species are distributed in the world. Like they get, like, you look at the map of South America, like it's really good. Like you see, like, near the forest you have a high number of species, and they are super old species. Of course, you're putting everything. You're simulating the world, so you get everything. So, but how can you understand, because things are changing. Okay, analyze the past is one thing. You want to be able to predict some things.
But this, you're not going to be, sorry, we can discuss this.
No,
it's not clear you're going to be able to predict something from this, right?
Yeah. Yeah, in fact, because tomorrow...
Yeah, I agree. So let's wait until tomorrow.
For example, one thing that we predict with this is the formation of ring species.
Okay.
It's a species that only makes sense because they are contiguous, like they are contiguous species, when in a contiguous space, but when we compare the extremities, they are not able to.
Yeah, I've heard
So you make a model that you can explain that without natural selection, for example.
Okay, so I agree with Silvina. Let's wait until tomorrow. And we'll
I
we'll
use
get
this
more
model
information.
to, for example,
I started by saying I'm not sure this is the right time to ask.
When I mentioned the fluctuations of the sea, I had the simulation, simulated the models when you have these things not continuous, but they fluctuate. And then you see you have explosion of species that they last very shortly and they die. So this, for example, would explain in some regions of the world that you have species that are very ephemeral.
Okay, very good. So let's hear about tomorrow. Okay, thanks, Deborah. Have any questions, right? Okay, so thanks, Deborah.